Hey, hello guys, welcome, vanakkam, vandanam, namaskaram. Today we are going to discuss such an important and a very easy lesson, that is molecular basis of inheritance. In this lesson, we are going to talk about those molecules which are being inherited by us from generations and generations. Okay, so already you guys would have guessed what it is. It is all just DNA. That's it. Okay, so and also just DNA. So this is actually one of my favorite creatures in this world. Okay, my best close friend, one of the most deepest trustable friend that you can ever have as a pet. So I have this as my pet. Okay, a very short tempered, very aggressive, and a very cute bird it is. So if you are hearing some kiki noises, then that is because this is what producing those kiki noises. So please just adjust those things. And the name of my little bird is Pikachu. Okay, it is also just as cute as Pikachu is. Okay, so coming to molecular basis of inheritance, what is inheritance? Inheritance is a process. Okay, it is just a process by which we are inheriting some traits from our parents to us. Okay, from the parental generation to the offspring generation. That is what we will be saying as inheritance. So this inheritance is actually because of some molecules. So those molecules are like DNA and also RNA, but DNA is the thing that you have to take. Okay, because DNA is the one and only substance. We will be seeing. Okay, why? Okay, so by what test did they prove that DNA is the genetic material? Why not RNA? All those things is we will be checking out. Okay, so before going into this process, you have to know what is central dogma. Central dogma is a process in which DNA express or a gene expresses its character. Okay, see, we all know that every particular trait that we have has some sort of gene. Okay, for an example, if I'm having this particular skin tone, then that is because of a gene that is expressing this particular skin tone. Okay, so that is why this color I have. Okay, so that gene will be expressing that one with some enzymes. So here I have melatonin, and melatonin has secreted in this particular amount. So that is why I'm having this particular skin color. If in case it has got secreted less now, then I will be whitish in color. If it had got secreted a lot now, then it will be blackish in color. So like that, it is actually because of DNAs. See, because of genes, I'll be just writing here. So there will be cell, right? Okay, wait. Okay, this I'm going to erase as it is not. It is very important, but you can just take note of it. There will be a cell, right? Okay. So inside that cell, what will be there? There will be nucleus. Okay, so inside nucleus what will be there? So inside nucleus there will be genetic material. Okay, so like we will be having the chromosomes. Okay, then you will be actually also having genes. Okay, then you will be also having, once again I'll be just saving my leg from my bird. No, my bird hates my leg for some reason, I don't know why. I do wash it regularly. Okay, coming to the topic. So gene. So from in gene there will be DNA. Okay. So this particular DNA is there, right? That we inherited by us from what? Okay, from where? From our parent only we will be inheriting this DNA. Okay. So this DNA should, okay, from this gene is the thing that is expressing the trait by using DNA. So how it is actually expressing, there will be DNA. So this DNA will be actually under, so this is what known as central dogma process. This is written there. Okay, this is written there. So by this is how it is actually done. So in central dogma process, okay, where DNA will be undergoing transcription and it will be forming RNA. So this RNA will be undergoing translation, which will be forming proteins. Proteins are made up of amino acids. At least, very minimum, at least 50 amino acids are required in order to form a protein. In order to form just a protein, 50 amino acids are required. So these proteins are those which are enzymes. We all know that enzymes are made up of proteins. Well, not completely. There are also coenzymes, all those cofactors, all these things are there, right? We have already studied them in biomolecules. So that's it. So amino acids. This is what known as central dogma process. So how our trait is being expressed. Okay. So coming to DNA. So see, I don't have that much time to take notes in the note. So I have taken notes in the tab itself. So We are just going to write here and we are going to see it. Okay, so DNA. See, DNA is a double-stranded chain. So what does it mean? It means it is just having two strands. Okay, it is just having two strands, which also means that just to form a DNA, all you want is, okay, all you want is two, okay, two what two? Two nucleotides. If you are having two nucleotides, then that is more than enough. Not more than enough, that is exactly enough to form, okay, in order to form a new DNA, okay, a DNA. Just these two are more than enough. It is enough. Okay, so the maximum amount, yeah. So first step, we will be coming step by step. Okay, next is that what is DNA? DNA is a long polymer of deoxyribonucleotides. What is this deoxyribonucleotides? Okay, see the deoxy word means from a compound, okay, from a X compound, okay, from this compound I have removed oxygen, okay, I have removed oxygen. Then there will be some remnant, right? There will be some residues, right? That particular remaining substance is what known as that. For that substance you have to add as prefix a deoxy. Okay, then here I am having ribo. Ribo, the word ribo means sugar, okay, pentose sugar. Here we are meaning it as pentose sugar. In DNA, there are totally three different substances, okay. So that is actually made up of, see, nucleotide is the thing that is full of DNA, okay. Nucleotide is the thing that creates DNA itself. So this nucleotide is having three different substances. First is going to be sugar, okay. So that sugar is what going to be the pentose sugar, 5-carbon sugar. Sorry, excuse me. Okay, so after this there is going to be a phosphate. We are going to see about it. Just a phosphate, that's it. There is nothing to see in this. Okay, but I don't know why I said we are going to see about it. Then we are going to have nitrogenous bases. What is this nitrogenous base? See, totally nitrogenous base is something that is very important. Okay, that is what is going to determine the nature itself. That is what is going to determine the total nature of the DNA itself. Okay. So this nitrogenous base is actually two different types. First will be purines and next will be pyrimidines. What is purine? Purine is a double ringed structure. Okay, so it will be like this. Okay, I'm going to erase it as it is not something that is in our book. In chemistry, we'll be studying very in detail what is purine, what is pyrimidine, okay, their structures, so what they have, what they don't have, what are their characters, how will be they reacting, all these. My English slang, there is some. And I got influenced by some of other persons, by some ex-persons. Okay, so I'll be having my own piece. Okay, see, this purine is there, right? So this purine is actually having two different substances. That is going to be adenine and then guanine. This pyrimidine is having three different substances. That is thymine, cytosine, and then uracil. Pyrimidine is single ringed and the purine is double ringed. Note this one point, Your Honor. That is, thymine is actually common for both. Yeah. Cytosine is common. Cytosine is common for both DNA and RNA. That means cytosine will be present in both DNA and then in RNA. But coming to thymine and uracil, they won't be like that. Thymine will be present in DNA, but it won't be present in RNA. Uracil will be present in RNA, but it won't be present in DNA. So this is something that you have to note it. Okay. So then after this, yeah. Base pair. See, as we normally measure something, for example, you can take this as a DNA, okay. If in case DNA were this big, then you can just measure them by using a scale. But since DNA is something that you cannot measure it with scale, we are measuring it with base pair. What is a base pair? Base pair is a structure, okay. It is a structure that is made up of two different nucleotides, okay. If there is just two different nucleotides, then that's it. That will be forming one base pair. Like that, we will be calculating how much base pairs are there in a DNA, and with that, we will be multiplying the distance between two base pairs. Okay, we'll be studying about that also. I'll be explaining it very cleanly. Just coming step by step. Then, in the structure of DNA, we'll be saying very keenly. Okay. Then the next thing is we are going to see about a number of base pairs. Okay, so we are going to see number of base pairs. So there is an organism. Normally, biologists are those who are a little bit different in thinking. They'll not just name a substance. For example, my name is Kannan. So that they won't name a bacteria or a virus known as Kannan. They'll be naming it with a different name. And so here there is a name as pi into 174. Okay, into 174. So this particular organism is actually having 5,386 nucleotides. 5,000, okay, 5,030, okay, 5,386, okay, 5,386 nucleotides. I cannot see clearly in this. These notes aren't that clear. Nucleotides. Okay, so this organism is having these many. Okay, so then what about remaining organisms? For an example, if you're taking here human, okay, human in haploid condition, a human is having 3.3 into 10 to the power of 9 base pairs, okay, base pairs. BP, not blood pressure, it is just base pair. Okay, the next is bacteriophage lambda. Lambda is represented by this symbol, right? Like this, they'll be representing. So that is what also I've drawn here. You have to accept it as lambda no matter what. Okay, because I'm that good in drawing. Okay, so it is having 48,502. I cannot see very clearly. 48,502 BPs are there. Okay, then a particular coli, okay, it's actually a coli variety. So coli, like E. coli, like that, there are some bacteria, okay, which is very good. They won't harm us. They're very soft. They're very good, like me. Okay, but I'm not a bacteria. They are a bacteria. And they are soft-hearted like me. So this particular name is kind of a little bit long. Okay, like my kindness. So it is actually Escherichia. Okay, it seems it is a Chinese bacteria. It has come from China, so that is why they have named it as like that. Okay, so that we all will be feeling very tough. So yeah, so Escherichia. Okay, chia. Okay, it has a 4.6 into 10 to the power of 6 base pairs. Okay, so the spelling alone I'll be reading of this. Okay, Escherichia. Coli, COLI, that's it. Okay, so that is what this particular Chinese bacteria is known as. We'll be keeping it as Chinese bacteria. Very easy, right? Okay, so next is a nucleotide chains. Okay. My bird is very naughty sometimes. It can be naughty. Okay, now next is we are going to see about nucleotide chains. Okay, so what is a nucleotide? I already have explained a little bit. Now we are going to see about the DNA structure. We are going to see about nucleotide, all those things is. Okay, so since for an example, we have to know how will be the structure of of purines and pyrimidines. I'm going to draw just two structures, okay, one of purine and one of pyrimidine. So I have chosen adenine. Okay, so adenine is belonging to what? It is belonging to purine. So its structure is going to be like this. First of all, it is actually having double ring. So first one ring you have to draw like this. Okay, so like this you have to draw. Then like this you have to draw. Okay, so see. This is how adenine will be looking like. Okay, then how will be pyrimidines? Okay, pyrimidines. How will be it looking? It will be looking like this. Okay, so I see here there will be nitrogen and then hydrogen. like this only you have to represent. If you are writing like this one, then it is wrong. You have to write it first nitrogen, then hydrogen only. It is because see here hydrogen is not connected to the carbon, it is actually connected to the nitrogen. Okay, it is actually connected to the nitrogen. Okay. Then after this, you are having here double bond oxygen. Then here also double bond oxygen. Then here nitrogen, hydrogen. Okay, so this is in pyrimidines. It belongs to uracil. Okay, it belongs to uracil. So this is pyrimidine. Okay, then this is purine. Okay, so that's it. So this is how the structure will be actually basically. Okay, so after this, coming to nitrogenous base pairing rule, which is actually also known as Chargaff rule. So now we'll be checking about it. Okay, actually this is something that you should have studied in class 11th itself. If you haven't studied, then this is for you. Papa, please. Thank you. No, please. Okay, which is also known as Chargaff rule. Okay, so it is also known as Chargaff rule, where see this is actually produced by Chargaff only. Irwin Chargaff is the person who has produced it, who had given it. Is the person who had given it. Oh god. Okay, so what he is, he said that one purine will be combining with one pyrimidine. Okay, one pyrimidine will be combining with one purine. Okay, the ratio of pyrimidine and purine will be always 1. Okay, that is what he said. So one purine will be combining with one pyrimidine. Some people also pronounce this as pyrimidine. Okay, you can pronounce it as your way. In a short, I'll be uploading how to pronounce it and check it out. Okay, how to pronounce it. Actually, it is like ஒருவருக்கு ஒருத்திக்கு ஒருவர், like that. Okay, for one girl, one boy, one girl, like that. You can remember it. Okay, so for one person, only one person can be soulmate. For one twin flame, only one will be the twin flame. Okay, there won't be 3 or 4 twin flames or yeah, like that. But soulmates can be a lot. Okay. So then after this, we are going to check about the connection between pyrin and pyrimidine. This connection is there, right? This connection can be of 2 different types. Okay, so this connection is actually of 2 different types. So one will be purine is there, right? So first, so when purine and pyrimidine is connecting, that connection is actually of 2 different types. One will be double hydrogen bond. Okay, then triple hydrogen bond. Okay, so this is what their connection is. Okay, there is no single hydrogen bond, double or triple hydrogen bond only. That means in this 1 pi bond and then 1 sigma bond, in this 2 pi bonds and 1 sigma bonds. Class 11th chemistry. Okay, you have to just input all the knowledge that you have into a particular place, so that you will be just revising. It will be always stored in your brain. Okay, so how it is? See, when adenine, no, I will use another color. Okay, so when adenine is actually combining with a thiamine, okay, so it will be having double bond. When cytosine is combining with the guanine is combining with the cytosine, it will be having triple bond. Okay, it will be having triple bond. And also same, when guanine is combining with when adenine is combining with uracil, it will be having double bond. Okay, see, note this. I hope you all know this, but if you don't know, you can just note it now. For RNA, okay, and then for DNA, the changes in nitrogenous bases are, see, in an RNA, what will be there? There will be adenine, there will be guanine. See, purines will never change. Only pyrimidines will be changing. In that pyrimidine also, cytosine will be constant. So what will be changing? Only this thymine and then uracil will be changing. For RNA, uracil is there, and then for DNA, thymine is there. Okay, other than that, all the components are the same. Papa, no. Come here. Bah. Good boy. Okay. Today he's listening to my words. I don't know why. Ouch, sorry. Okay. So this is what you have to know in this Chargaff rule. And also one more thing you have to know, that is, I don't think that is Chargaff rule. So some other concepts that you have to know is, you can take this as equal to sign. Always adenine will be equal to thymine, guanine will be equal to cytosine, adenine will be equal to uracil. Okay, so these things as you can take it as. Adenine and then thymine will be equal, adenine and then uracil will be equal, guanine and cytosine will be equal. Okay, so this is something that you have to know. So you can also take it as, when you are adding all these compounds, for an example, when you are adding all the compounds in DNA, that means adenine plus guanine plus cytosine plus thymine, it will be equal to 100%. For an example, okay, we'll be just doing a particular sum. One sum we'll be doing, that's more than enough. They have given the percentage of adenine. Then you can find the remaining percentages. Very simple, very easy. So, since they have given the percentage of adenine, let us keep that as 20%. Okay. So, what did I say? Adenine and thymine will be equal. So, thymine will be also 20%. Okay. So, see, guanine and cytosine will be equal. So, that means G is equal to C. That means I can put in place of here just 2C I can put, right, is equal to 100. Is equal to 100 like this. I will erase it. It is not, yeah, it is very important. But wait, then I'm trying here. So it is 20%. So this thymine will be also 20%. And the only thing that you have to know is that guanine and cytosine are equal. So you can say it as 2C, which is equal to 100%. Okay. So here it is actually 2C plus 40%. Sorry, plus 40% is equal to 100%. So which is equal to 60%. Where 2C is equal to 60%. So C is equal to 30%. So, which means guanine, okay, so guanine and cytosine are equal. So, guanine is also having 30%, cytosine is also having 30%. Yeah. So, I can rewrite it as, so guanine is equal to cytosine is equal to 30%. Okay, so is equal to 30%. Tada, you got it. Okay, so then the next thing that you have to know is, now we are going to check about what else shall we do. Okay, first we'll be completing all the basic topics that we have to discuss. Then we'll be coming to the structure of DNA. Then with the structure of DNA, all those data that are required, all these things, we will be completing this video itself, the total video. In the next part, we'll be seeing very clearly about, okay, about 40 minutes one video is done, and then that is enough for me. Per day, about 80 minutes if I'm filming, that's okay for me. So then about pentose sugar shall we see. About pentose sugar shall we see. As this can be asked as 1 mark questions in MCQs, they can ask it. I think also NEET question one time they asked. I don't know whether it's correct or not. If I'm not mistaken, it is asked a NEET question one time. Okay. So now coming to pentose sugar. What is there in this pentose sugar? It is just a sugar with 5 carbons, right? Well, the thing is, this sugar differs. The sugar differs with respect to DNA, okay, with DNA and then with RNA. The only difference is that DNA does not have this oxygen, but RNA has this oxygen. In the second carbon, okay, in the second carbon, it is actually having this oxygen, and here it is not having. So how will be the structure then? Okay, see, first of all you have to do is first you have to draw a pentose, okay, a pentagon. Wait, shape. Okay, perfect, I got it. Oh. Okay, so here, see, now you have to start to name. So before naming, you have to put your O. Here also you have to put your O, which represents oxygen. Okay, so now we have to start numbering. 1, 2, 3, 4, and then here will be fifth carbon. Okay, 1, 2, 3, 4, and then here will be fifth carbon. Like this, a few people will be drawing, and also like this, also a few people will be drawing. 4, 5. 4, 5. Okay, so but I prefer like this only. Okay, so like this only I'll be preferring. So I'll be also drawing with that one. Okay, see, this RNA is actually having here OH. Okay, here all it is having is just H. That is one difference that you have to note. Okay, so this oxygen is there, right? This oxygen that is in RNA, right? That is very, very reactive. That is highly reactive. Very, very reactive. That is making this RNA as less stable. So that is making this RNA to pawn to mutation. Okay, that is making this DNA, since here it is not having that oxygen, it is making this DNA as more stable, so that DNA cannot be mutated. Okay, so then after that, in the fourth carbon is in the fifth carbon is there, right? So in the fifth carbon there is CH2OH. Here also there is CH2OH. Papa, come here. Come here. Bah. Good boy. Okay, so then after this, the first carbon, there is actually OH. Here also OH is there. Okay. Again and again you are going there. Stay there. Okay, so still you are there, I don't have any problem. Deoxyribonucleic acid pentose sugar structure. Hydrogen will be here. Hydrogen will be here. Pentose sugar of DNA. Yeah. So here, here OH will be there. Here also OH will be there. Only here, only the difference will be. Okay. So here in this notes, there is actually a small mistake. We will redraw it. Okay. So yeah, that's the difference. I got confused with that. Okay. So this is how the pentose sugar will be looking. Stay right there. And also please comment about my bird. How obedient is it? How have I grown it? I was the one who fed it up. Very proud, okay? I know I'm kind of flexing it out, but very, very proud to grow such a pleasant, short-tempered, very angry creature. Okay, when it sees a stranger, that's it, it kills that person. Okay, so now coming to nomenclature. See, for nucleoside and for nucleotide, you are having different nomenclatures. We'll be checking about them. So, what is a nucleoside? Nucleoside is something when you add nitrogenous base to the pentose sugar, it forms nucleoside, and to that nucleoside, if you are adding phosphate, then that forms nucleotide. That's the difference. Okay, so now let us see about it. Okay, so first is nucleoside. So what is it is actually what it is just a nitrogen base plus, some people also say it as nitrogenous base, nitrogen, but we will just say it as nitrogen base. Okay, nitrogen base plus sugar. Okay, so how is their nomenclature going to be? It's actually very simple. When you're adding guanine plus sugar, okay, plus sugar, it gives you guanosine. Sine, okay. So when you're adding adenine, okay, when you're adding adenine plus sugar, okay, then we'll be giving you adenosine. Okay, then we'll be giving you adenosine. But this is actually applicable only to purines. Coming to pyrimidines, it is actually kind of different. Thiamine. Oh shit. Oh, come here. Bah. Bah, bah, bah. Bah. Thiamine plus sugar. No bitings. Papa. Okay. Uracil plus sugar. Okay, I'll just keep it. Yeah, it's actually kind of late now. Okay. So when you're adding uracil with sugar, and then when you're adding cytosine, it gives you thymidine. Okay, it gives you uracimidine, then it gives you cytomidine. Yeah, uracimidine, thymidine, and cytomidine. Okay, here it gives you uracimidine. Then here it gives you cytomidine. Okay, this is what it actually gives to you. Okay, so yeah, this is what urimidine, adenosine and urimidine. Okay, next is when you are adding phosphate to this. Okay, so nucleotide. Okay, so nucleotide. So how is nucleotide formed? It is formed by when you are adding nucleoside, and with this nucleoside, if you are adding phosphate group, then this forms a nucleotide. So how is this actually working? See, guanosine to guanosine, when you are adding phosphate, then this gives you guanilic acid. Okay, so to adenine, okay, sorry, to adenosine. When you are adding phosphate, it gives you phosphoric acid. Okay, it gives you, sorry, adenolic acid. Okay, it gives you adenolic acid. Okay, then to thiamidine. Okay, to thiamidine. When I add phosphate, then it gives you thiamelic. Okay, so thiamelic acid. So when I add to uracimidine, phosphate. Then here it gives me uracilic acid. So when I give phosphate to cytomidin, that's the remaining thing, right? Cytomidin plus phosphate. Okay, so it gives me cytolic acid. Cytolic acid. Yeah, that's it. Well, here it is actually D will be coming. Thiamidilic, uracilic, cytomidilic, cyt. Okay, so this is it. So cytomidilic acid. Okay. So how will you actually draw this structure? Okay, so how will you draw this structure? This question is something that is very, very, very important thing. Okay, because this is how you are going to represent it in your examinations. Okay, so before that, we are going to see about a few things. That is going to be helix. See, normally this is how the DNA will be. Okay, wow, I'm very impressed. Okay, so like this only DNA will be. So in this one helix, okay, so if you are considering one helix, so you have to take half of this helix and half of this helix. So then this will be one helix. Okay, so this will be one helix. And in this, if you are considering this one, this is known as upper guru. This is known as lower guru. So it's actually not guru, it is actually known as groove. Upper groove. Okay, here it is lower groove. Groove. So upper groove, lower groove, so this part and then this part. That's it, very simple. Okay. So now the thing is, now another thing that we have to see is, normally see, since we cannot just each and every time we cannot draw each phosphate group, we cannot draw that hydrogen bond, we cannot draw the phosphodiester bond, we cannot draw sugars, we cannot draw purines, we cannot draw pyrimidines each and every time. What people will be doing is, they'll be considering this as a strand, and they'll be just writing here those pyrines and pyrimidines. Like for example, here adenine, here thymine, they will fill. Here guanine, here uracil, they will fill. Like that, they will just fill it up like this, and then they will be going. Okay. So here these. Okay. So here this one is there, right? So here this will be considered as 5 dash. This end will be considered as 5 dash. Here this end will be considered as 3 dash. Here this end will be considered as 3 dash. Here this end will be considered as 5 dash. Okay. So here this is what known as a replicating template. Okay. So because not complementary, okay, it is actually complementary, because here, okay, see in this if your adenine is there, here automatically thymine will be there. So whatever purine or pyrimidine is here, the exact complementary purine or pyrimidine will be here. Okay, so what is that exact complementary or complementary purine or pyrimidine? See, this is what it is. We have already seen, right? That is what. Okay, so how are you going to do with this? Okay, how are you going to draw this one? See, you can draw it vertically or you can draw it in a horizontal way. So we will be now drawing about it. Okay, before that thing, once again. Okay, so this one is there, right? So here I have to say a few things, just a few things. Okay. Okay, so here, this is how the DNA will be. Okay, this is how the DNA is going to look like. One second. Nope, I don't have it. So see, the thing that you have to remember is this total turn is there, right? So first in the very beginning, it will be like this. Okay, it will be like this. Then in the next, see this each line that I have drawn here, right? This line, this line, this line. This represents one base pair. So one base pair now, this particular base pair will be having one purine and then one pyrimidine. Pyrimidine. You can say it as pyrimidine or pyrimidine. Okay, pyramidine. So one purine and one pyrimidine will be having. So this will be consisting one base pair like this. Within this particular distance, okay, within this one helix you can take. I have said now this half and then this half is what known as one helix, or you can take this one also. Okay. Actually, the thing is one helix should have this one turn, this one pitch. Okay, this should be this connecting point. This should be there. That is why we are considering from this to this as a one helix. Okay. So that one helix will be having around 10 BPs. Okay, we'll be having 10 BPs. Okay, and one helix will be completely rotating into 360 degrees. So that means if the first BP is like this, then the second BP will be like this, then like this, then like this, then like this, then like this. Each and every base pair will be having 10 degrees as their difference. Like this, they'll be rotating. At last, if the first is like this, if the first base pair is like this, now then the second base pair will be like this. Exactly 360 degrees. Okay, exactly 360 degrees will be turned. Okay. In between 10 base pairs, there will be 360 degrees turn. Each and every base pair will be having 36 degrees turn. Okay, 36 degrees of turn. Okay, then exactly the distance between, okay, the distance between two base pairs. Okay, if you're taking the distance between two base pairs, all of this I'm going to draw it in a very neat way. I'll be first explaining this one, then you can understand while I'm drawing. Okay, it'll be like a revision to you. Okay, so the distance between these two base pairs, okay, like from this point to this point is there, right? So that will be 3.4. What is that? Not nanometer, Armstrong. Okay, so it's going to be 3.4. Or I can take it as 0. Okay, so 0.34 nanometer. Okay, 0.34 nanometer or 3.4 Armstrong I can take it as. So what is Armstrong? Armstrong is 10 to the power of minus 10. Nanometer is 10 to the power of minus 9. Okay, so the distance, so distance between these two is what? 0.34 nanometer. So then what will be the distance from here to here? All you have to do is you have to multiply it with 10. You have to multiply it with 10. So if you are multiplying it with 10, then how it will be? It will be like, okay, so it will be 3.4 nanometers. That means the total distance, okay, so of one helix. So if you are considering this part to this part, it will be exactly 3.4 nanometers. Okay, but the distance between two base pairs will be only 0.34 nanometers. Okay, then the distance between two terminals of base pairs, that means this point and then this point. Okay, so there are these 2 points, right? So this distance is actually known as 20 nanometer. Okay, so this point is actually 20 nanometer. Okay, 1 second. Okay, so to just, I'm very weak in technology. Okay, so to check, okay, so to read it, I have taken it. Okay, I'm using a lot of okay, right? So this is how the DNA structure is going to be. Now we will check it out very clearly. Instead of me drawing, even no matter how many hours I spend, I cannot draw like this, right? So I've got it. See the distance. The distance. First of all, we will be checking out very clearly each and everything. So this groove, we will term this as upper groove. Okay. This groove, we will term it as lower groove. Okay, then after this, we will be considering one helix from here only. Okay, from here to here only, we will be considering this as one helix. Okay, so this one helix will be having 10 base pairs. Okay, first of all, their angle. Okay, so angle between every base pair is going to be 10 degrees. So the total angle between one helix, okay, total angle of one helix is going to be 360 degrees, as already said. Okay, then after this, the distance between two base pairs. Okay, distance between two base pair is going to be just this distance net is going to be 0.34 nanometers. Okay, so what is nanometer? So 1 nanometer is equal to 10 to the power of minus 9 meter. 1 Armstrong is equal to 10 to the power of minus 10 meters. Okay, this is something that you have to know. So distance between sorry. So two base pairs is going to be this one. Then one helix. Okay, one helix distance. Helix distance. Okay. So distance I'll be representing as dtw. So that is going to be, what is that dtw? One helix distance is going to be 3.4 nanometers. Okay, 3.4 nanometers, because you have to multiply here with 10. Okay, you have to multiply there with 10. Then the distance between 5 dash end and then 3 dash end. That means, see, first of all, the distance between two base pairs now what? So if you are considering this as a base pair, and if you are considering this as a base pair, then this distance is what? 0.34 nanometers. Okay, and one total helix. So that means this total distance from this total distance is actually going to be 3.4 nanometers. Okay, nanometers. Then this distance from this to this point, okay, there will be some distance now, so you can also take that as the length of one base pair. So from this point to, okay, so from this point to this point, if you're taking that distance now, then that distance is going to be 2 nanometers, or 20 Armstrongs also you can take. Okay, or 20 Armstrongs also you can take. Here also you can take it as 34 Armstrongs. Here you can take it as 3.4 Armstrongs. Okay, yeah, that's it. So see, here as I said, right, so here is the place where the sugar will be there, and the sugar will be connected to phosphate. Here also one phosphate will be there. Sorry, here phosphate, no, there won't be any phosphate. There phosphate won't be there. So this will be one nitrogenous base. This also, sorry, this is going to be one purine, and if this is a purine, then this will be a pyrimidine. Okay, so this is going to be one end, and then this is also going to be another end. So that is what you have to know in this space. One end. So if I'm taking this as 5 dash, this is going to be 3 dash. Then this end is going to be 5 dash, and then this end is going to be 3 dash. Okay, so this is cytosine. So for cytosine, you will be having guanine. Yes, correct. Sorry. Yes, cytosine and guanine will be connected. See here, adenine, this particular green color is there, right? So this is adenine. So for adenine, you will be having thiamine. So this is going to be thiamine. Like that, you have to check it out. So I hope it's all clear. I have given it. And also, what is the backbone? Okay, backbone is going to be sugar and phosphate is what is the backbone. Okay, so sugar and phosphate is the backbone of a DNA. This strand is what the backbone of DNA. Okay. So this is how you have to, okay. Now we are going to draw the structure by our own hand. Okay, if there is again a one shot, the total one shot video is there. This is part 1, I'm going to upload. So if in case there is total one shot video, now then I will again give a seminar like thing. This is also a seminar. Okay, now DNA structure. Okay, so I think we are covering very slowly, but we are doing our level best. And also one more thing I forgot to say. See here, even I've said, right, human is actually having a 3.3 into 10 to the power of 9 base pairs in haploid. Then in diploid condition, then how many will be there? 6.6 will be there. Okay, so in diploid 6.6 into 10 to the power of 9 base pairs will be there. Okay, so here the total length of human DNA, if you're taking just the total length. See, so how many base pairs are there in human? If you're taking the total length, 0.6 into 10 to the power of 9 is there, right? Okay, so this is what the total base pairs are present, and the distance between two base pairs is what it is. If you are taking here, and now since here 9 is there, so I will take nanometers, which is 10 to the power of minus 9. So it will be 0.34 into 10 to the power of minus 9. So this will be cancelled, this will be also cancelled. So if I cancelling these two terms, it will be 2.2 meters. So the total length of our human DNA is going to be 2.2 meters. Then just imagine such a long thread, such a long thread. The length is 2.2 meters. A thread whose length is 2.2 meter is being coiled and coiled and coiled and coiled and coiled, folded and folded and folded and folded, and then it is kept into a cell. Then just imagine how great the biology is, okay, how great the system is. Isn't that very interesting? Okay, so now what we are going to do is we are going to draw the diagram. We are going to draw and see the diagram. Okay, first of all you have to, now I will be going with the NCERT book model itself. So since we are reading NCERT, it's our first priority. So first we'll be going with NCERT itself. First the sugar will be there. Everything will be white. Okay, everything I will draw in white. So this is what a pentose sugar is going to be. Okay, so since we are going to draw DNA, the one now here oxygen will be there. Okay, so here here CH2 OH will be there. Okay, then here OH will be there. Here H will be there. Here also OH will be there. Okay, for a ribose sugar. So like this itself. See here even we have already drawn. Okay, here OH, here also OH will be there, right? So here also OH, like this only it will be. Okay, so like this, exactly like this itself. Oh shit. So exactly like this, another will be joined. Okay, another will be joined here. Okay, so exactly like this, another will be joined. So when that is joining, all you have to know is, okay, you have to know one point, that is going to be this OH will be removed. Okay, this OH will be removed when another sugar is being added to here. This OH will be removed, and then there will be H. So this point only will be joining here. Okay, so it will be inverted. Okay, it will be exactly inverted. Wait, I will draw it very clearly. Okay, so here to this point, this another sugar of this hydrogen will be there. So this will be there. So these two will be going out as H2O molecule. This will be gone out. Then only this the total process will be happening. Okay. See, even I have no experience in this. We'll be drawing it very slowly. Okay. The way I have practiced, even I will be drawing. All you have to do is you have to just tilt the diagram. Okay. So it's going to be, see, first here this is first carbon, this is second carbon, this is third carbon, and then this is fourth carbon. Okay, so to this carbon there will be here in this case, okay, this is just a sugar molecule. If it is just a sugar molecule, it will be like this, okay, but since it is in the DNA, there will be some change in it. Okay, some phosphates will be added, some other things will be added. See, to this sugar molecule, guanine is being added. So the structure will be pentose only, but the component like this OH, like the CH2 will be removed from that, and then there will be some change. There will be some changes. So those changes will be like this. See, here in this place, a carbon will be there. So here hydrogen will be there. Here also hydrogen will be there. Then here phosphate will be there. Okay, so if I am taking this as a 5 dash end, yeah, so this is actually fourth carbon will be present here, fifth carbon, okay, 5 dash only will be there. Okay, so 5 dash only will be here only fourth carbon, here oxygen is there. Okay, so here this is actually a second, so second will be having only hydrogen, here will be having OH. So this is how this sugar will be there in a DNA. Okay, so to this sugar in this place, you have to take it out, and instead of there, you have to put a nitrogen base. Okay, so here, if I'm adding here thiamine, if I'm taking here thiamine, okay, so this thiamine will be having double bond with what? With adenine. Okay, with adenine. Normally, you can draw like this, or also you can take it as like the enzyme we'll be putting up for enzymes. Okay, here double bond is there. Okay, here double bond will be there. So to this adenine part, okay, so to this adenine part, exactly the inverted of the sugar molecule will be here. So that means oxygen will be here. So here, if okay, see, here one will be there, here one is there, exactly like this. If you are putting that, so here one will be there, so here two will be there, here three will be there, and then here four will be there. So this four, there will be five here. Okay, so if 5 dash is here, so that means what? If 5 dash is here this fifth carbon only we are saying it as a 5 dash. So here this third carbon only is represented as 3 dash. Okay, so note that point if you are having confusion, and then so this is how this came. Okay, so here one phosphate will be there. Okay, so here one phosphate will be there. So to this phosphate also, here see in this place, okay, so till now we will be just checking. Okay, so here in this 5 dash end, one phosphate will be there. Here also one phosphate will be there. So here double bond will be there. And the distance between this one, I said it as 20 Armstrongs. Okay, or 2 nanometers. Okay, or this is what I've said as 2 nanometers. Okay. The bond which is here is actually a hydrogen bond. And then the bond that is present here, right? There are two bonds. See, in this one BP itself, there are nitrogen. Sorry, there are two what nucleotides. So in that two nucleotides, each nucleotide will be having one N glycosidic bond. Okay, one N glyco. So these two are two. Okay, so since there are two, so I have represented here two. Oh, no, just I will write N glycosidic bond. Okay, N glycosidic bond. Okay, N glycosidic bond, and then this bond is actually known as hydrogen bond. Okay, so this bond is known as hydrogen bond. Here hydrogen bond is 2, here also hydrogen bond is 2. Okay, now what will be happening is here phosphate is there now. So like this itself, there will be another sugar molecule which will be tilted. Okay, here and here there will be okay, so here will be see like this. Okay, like this only it will be. Actually here this is connected to 1. Here also it is actually connected to 1. Here it is connected to 1. Same thing. Here also it is connected to 1. Here also it is connected to 1. So do not let that point get out of your mind. Okay. So here if I am putting guanine, so then here will be cytosine. Here there will be triple bond. Okay. So here will be first carbon, here will be second carbon, here will be third carbon, and then here will be fourth carbon. Absolutely yes, right. Okay. So here in this third carbon there will be OH. Okay. So in this fourth carbon, see what's there in fourth carbon? In the fourth carbon you are having CH2OH. So here also you are having CH2OH, right? So in this carbon, okay, in this third carbon you are having OH. So from this, okay, so from see first there will be OH, sorry, yeah, CH2OH will be there. First CH2OH will be there in this fourth carbon. Okay, so this will be combining with this. Okay, it will be forming CH2 and some other product, and then it will be gone. Okay, and then it will be gone. So after that it is gone, there will be a bond will be formed between here. Okay, so to this phosphate and this phosphate, the exact same thing will be happening here. If you are naming this as 1, 2, 3, 4, and then fifth carbon, so here also this OH will be removed. So here will be a phosphate, here also will be phosphate. Okay, so the bond between two base pairs, this is a base pair, this is a base pair, right? So to connect these two base pairs, what we are using, we are having the here also, see here also, there are two. What are there? There are two. Okay, these bonds are known as ester bonds. Okay, so there is two phospho. So since there is phosphate, so phospho phosphodiester bond. Phosphodiester bond. So if you want to represent it in this one, see here, okay, so this point and then this point is there, right? So these two are connected. In this between there will be phosphate, okay? So this is actually phosphodiester bond. What is here? So in this place a hydrogen bond will be there. In this place a hydrogen bond will be there. In this place will be a hydrogen bond will be there. Then what about this N-glycosidic bond? N-glycosidic bond will be between this. What is this? This is actually thiamine. So this red color is thiamine. So this red color, okay, this thiamine, and then to the sugar molecule, okay, to the sugar molecule will be N-glycosidic bond. That's it. So we have got completed with this one. Thank you. We will meet in another video. Bye bye.